Okay, I'd like to welcome people back to day 2 of our workshop. Today, I'm going to be talking about some examples of what you can do with a program like CompuCell, and then I'm going to talk about some principles of modeling. Those may seem a little bit abstract, but I hope, especially for people who want to do the hackathon, that they're going to be useful. This is going to be a bit of a lightning tour because this is actually now 2 modules that are being compressed into one. And I'm happy to go over these materials more later with people. And there'll be additional slides that we probably won't cover that will be available for people to look at in the student materials. As always, I need to remind you that the workshop is live streamed, recorded, and distributed. And I'll say that this is more of a lecture. We typically try to avoid having blocks of lecture without a lot of student exercises, but this is going to be more of a lecture. So today I'm hoping we can talk a little bit about some of the possibilities and opportunities of using this kind of model. I'll show you some examples from the past 10 years of work that was done using it. And then we're going to talk to see about how you have to think about building models. Because in my experience, while building the software implementations that we've been doing in this class is challenging, actually the hardest part of model building is to think about the problem and defining the problem. So if we want to know why we need to build multicellular models, I come from bioengineering, and in the world of engineered systems, everything that we do is designed on a computer. And not only is it designed on a computer, there are computer simulations of how it works. And so we typically know how engineered systems are going to work before they're released into the wild. Now, they don't always work the way we think they will because our computer simulations aren't perfect. But nevertheless, most things that we encounter that are manufactured are designed. And biomedicine is about the one thing in the world that's not engineered. And the reason for that, of course, is that we don't have blueprints and specifications for the human body. And so there's a lot of science that we don't know about how it works. One problem that we run into is that medical treatment, even with the current terminology of precision medicine and personalized medicine, is still really based on population averages. It's not optimal for any individual. Now, there are teams that are using the kind of modeling that we're talking about here, not our particular software, but similar software, for example, at Moffitt Cancer Institute at Tampa, that are designing truly personalized cancer treatments. So it's moving in that direction, but we still need to come up with personalization. The modeling we're going to do this week isn't going to get us that far for that, but these are methods that in principle could be used for building truly personalized predictive medicine. Something that you come up with in medicine a lot is that you can't repeat an experiment to determine best treatment. You can't treat and not treat the same patient. And I had this experience the other day. I had COVID 2 weeks ago. I took Paxlovid. My friends had COVID at the same time, didn't take Paxlovid. They recovered more rapidly than I did. I have no way of knowing whether the Paxlovid helped me, and I would have been even more sick if I hadn't taken it, or whether somehow the Paxlovid actually slowed down my recovery, because there's no way I could do the experiment to know what would have happened to me personally if I hadn't taken the medicine. So that's an example of the kind of issue that we might face that we want to address with models. We can't do control experiments clinically either, really. That's another problem. We often don't know what the cause of a disease is, and that makes it harder to design therapies for it. And something that is a dream of mine would be to make medical care proactive rather than reactive. You don't wait for problems to happen. Have closed loop medical. Again, this is moving more in the direction of large-scale bioengineering, but I think the things we're doing here are a piece of that. Clearly, if we're going to do control of biomedical processes, human health, we have to be able to predict. And often that terminology these days comes up with the word digital twin. A digital twin is often misused. I'm not going to go into all the details of what digital twin really is today, but I think that the kind of modeling we're doing here is potentially a component of building digital. If we can build models, whether those are models of in vitro systems, organoid culture, animals, or humans, we can do a lot of things that are impossible to do otherwise. In particular, you can run unlimited numbers of replica experiments. You could try out all sorts of possible treatment strategies. You can come up with strategies that are optimal for individuals, and you can also do scientific discovery. And so I think there are a lot of opportunities here, both in basic science, applied science, and medicine, to use these methodologies. If we ask how much are these kinds of methods used today, the sort of multicellular modeling that we're talking about here is still not used very much in biomedical industry. It's mainly used in scientific discovery at this point. But if you go to other scales, there is a lot of predictive modeling that's being used for personalization of medicine. Probably the most successful company at the moment in this area is a group called HeartFlow that was out of Stanford that does continuum mechanics modeling of major blood vessels and blood flow. So this is computational fluid dynamics and solids modeling to diagnose and design treatments for aneurysms. And they just raised almost $500 million a few months ago in their commercialization. There's nothing comparable to that in the multicellular world that we're working in, but perhaps one of you will be the one to do that in the next couple of years. We've talked about AI and mechanistic whole body digital twins a little bit. Molecular dynamics, of course, is used in rational drug design a lot, and that is an established component of modern drug discovery. And another area where multi-scale modeling is used, typically not, again, modeling tissue so much, but a very interesting opportunity, are virtual clinical trials. There's a company out of Lyon, France called Nova Discovery that's doing really interesting work in that area. And some of the people who've worked on CompuCell actually have gone to work for them afterwards. But again, this kind of virtual tissue modeling that we're doing with cells as our active agents is just beginning to find medical applications. The Moffitt people, I think, are an example of that. There are others. And I think it's important to remember that these kinds of approaches are complementary to omics, AI, continuum modeling, in vitro methods. Ultimately, we want to put them all together. So if we look at CC3D, here are the screenshot, probably a little bit out of date now, a screenshot of some recent papers published using CompuCell. You see that there are quite a few papers related to cancer and other papers related to infectious diseases. There are a few papers related to basic biology and development. And so there are a variety of different applications that one can apply these methods to do. If we look at cancer and developmental diseases, a classic work that we did a long, long time ago, which is a beautiful simulation, we've seen it many times because I think it's aesthetically very pleasing, is this model of a tumor spheroid. We have a tumor that grows, it forms a quiescent layer and then a necrotic core. It's receiving oxygen and glucose from pre-existing blood vessels. When it becomes hypoxic, it sends out a vascular growth factor that induces neovascularization. And we can compare a situation without neovascularization on the right, neovascularization on the left. Otherwise, everything is the same. And we can ask, how does the tumor grow as a function of time? In both cases, the tumor grows. The domains of growth are actually not that different. Early on, the tumor growth is identical. It takes a while for things to diverge. And we could ask the question, what are those regions of development of the tumor? What's the effect of the tumor vascular direction? We would have probably should have continued that work much more. But one of the things one could have done, for example, would have been to explore the effect of anti-angiogenics on the tumor. We've done a lot of work with Bob Bacalhau, MD, kidney surgeon at IU School of Medicine, who works especially on a disease called autosomal dominant polycystic kidney disease. This is a proliferative disease. Like cancer, cells grow inappropriately. Unlike cancer, you don't typically have progression and metastasis. Cancer is often thought of as a genetic disease, but in fact, there are relatively few genes that are mutated in most cancers. APC, P53, RAC maybe. But in general, cancer is not a genetic disease. It's a disease of cell behaviors. And the reason for that is that there are many different ways you could have the same changes in cell behavior. There aren't unique genes that are responsible for cell motility, for example. P53 is a little bit special because apoptosis really is channeled through P53 for the most part. In the case of autosomal dominant polycystic kidney disease, there are two genes, polycystin 1 and polycystin 2, that are mutated essentially in everyone who gets the disease. If you have a germline mutation in those two genes, you are essentially going to get the disease. If you don't have a germline mutation in one of them, your probability of getting it is 10 to the minus 6, 10 to the minus 7. They are ciliary proteins and associated with ciliary proteins. How that affects proliferation is not well understood. Drugs that targeted them directly didn't work. Our collaborator hypothesized that the issue might be that PKD1 and PKD2 associate with cell adhesion molecules. And so the question was, is it possible that what's happening in this overgrowth of the tumor, overgrowth is the kidney, where the kidney develops cysts and continues to grow throughout life, is related to an adhesion defect. And Giulio Belmonte, who's lectured yesterday, will be lecturing more, built a simulation of kidney tubules in vitro. In this case, you take a kidney tubule that has a set of contact inhibitory signals. Those contact inhibitory signals lead to a stable tubule. You imagine an experiment where you transfect a single cell with an ectopic cadherin, in this case, cadherin 8, which is a juvenile cadherin. And we made various hypotheses about what that cadherin could be doing. It could be changing cell-cell adhesion directly. It could be changing the mediation of contact inhibition or some combination of the two. If you have it change adhesion directly, you get a cystic phenotype, which is what's shown here in the video. The cystic phenotype also occurs with decreased contact inhibition, but the time evolution and the morphology are different. Bob went into an in vitro HK1 cell line, transfected it with cadherin 8, ectopic cadherin 8, and was able to show that you get cysts of the same morphology that you get in the experiment simulation. Every cell in the cyst expressed the ectopic cadherin. Every cell with the ectopic cadherin was in the cyst, so it was a one-to-one map. That was actually very nice. A little bit unusual that you get that clear a signal. Then there's still a question about how the cadherin is working. Cadherins modulate cell adhesion and cell motility through beta-catenin. Beta-catenin in turn connects to cell cycle through phosphodiesterases. And so this suggested particular classes of phosphodiesterase expressed cadherin and excess proliferation. And we've been doing some animal studies of using phosphodiesterase inhibitors. Ironically, they're drugs that are pretty common anyway, sildenafil and tadalafil, which are otherwise known as Cialis and Viagra, as treatments. They work well in mice. Of course, showing that they work in mice doesn't show that they work in humans. And so Bob is currently in preclinical animal studies of this. We've done a lot of work over the years, and CompuCell was originally really developed to do developmental biology. You've seen this picture before. This is a chicken embryo developing, forming the midline structures that are going to lead to the vertebra and ribs, the somites. You've seen this picture as sort of a basics for the understanding of the multi-layer structure of virtual tissue models. In this case, we have a molecular clock simulation of a variety of oscillating genes, FGF, Wnt, and Notch oscillations. Notch couples with neighboring cells, so it's got a juxtacrine mechanism. There's a model of cell proliferation and also cell differentiation. That cell differentiation in turn, in conjunction with the clock, leads to particular patterns of cell adhesion molecules, both normal things like cadherins and CAMs, and heterotypic molecules like F and F1, leading to this. What was interesting, and this was again work with Giulio Belmonte and our collaborators in London, Claudio Stern and his students, was that Claudio was able to show that you could actually turn off the clock and the wavefront in that model, in the experiment, and still produce somatic structures. And so Giulio then did a self-organization model. People were asking about compartmental cells and elongation. These are models of cells that are starting out essentially as mesenchyme. They're going to polarize. You'll see the red and the green compartments. They're going to elongate, and there is a mechanism, a contact mechanism that co-aligns the polarization, leading to the formation of clusters and somites. We're going to hear from Priyam Ajapok, who took that model a little bit further. Here we have an epithelial layer along the midline. We have apical contraction of actin. That's leading to mechanical changes and the separation of the initially contiguous tissue into patches, which are something that you see during early development. This is another model of Giulio's. This is going into subcellular detail now. He was interested in the propagation of hair cell orientation in imaginal discs in Drosophila wing. These were experiments originally done and published, very famous experiments by Axelrod at Stanford, who studied mutation patterns that lead to disorganization of the hair orientation. The genes have wonderful names like frizzled and Van Gogh and flamingo. Van Gogh, because if you interfere with Van Gogh, the hairs are still locally oriented, but they form spirals, and somebody thought that looked like the Starry Night painting of Van Gogh. In this case, Giulio has modeled the subcellular location of the membrane proteins that are involved in planar polarity and their induction. And so somebody had asked about modeling subcellular organization. The last two slides show examples of modeling subcellular organization. We've worked quite a bit on retinal vascular diseases with CompuCell. I'm only going to show one example here. We've been interested in wet age-related macular degeneration, which leads to neovascularization, where the blood vessels behind the retina invade the retina and eventually cause it to detach and deteriorate. The classic hypothesis here was that this was related to excess growth factors. We, as usual, as you'll find out, adhesion is very important in our world, thought that adhesion was probably an issue. And Abbas was working with a lot of ex vivo tissue samples. And he noticed, actually courtesy of a bad shipping by FedEx, that it was easier to have the retina break up in patients with AMD. These are deceased patients. These are eyes from eye banks. Than it was in retinas from patients which had died without age-related macular degeneration. And so his hypothesis was that actually it was an adhesion defect between the pigmented retinal epithelial cells and the Brooks membrane at the back of the retina. And he built a series of simulations of the retinal structure here in 3D. You have the vasculature behind the retina, the retinal pigmented epithelium, the black lining of the eye, then the photoreceptors on top. Here, green is pigmented retinal epithelium, purple is blood vessels, gray is photoreceptors. The color scheme here is a little bit different. Here we don't show the photoreceptors, and the neovasculature is in red. Here, create a hole in Brooks membrane. Under normal conditions, the adhesion environment is such that the blood vessels can't penetrate. They always try to because there's growth factor in the eye, because the eye is always hypoxic. But in the case of an adhesion defect between the pigmented retinal epithelium and Brooks membrane, and that happens naturally as we age, the waste products from the eye have to cross Brooks membrane. Lipids accumulate on Brooks membrane and reduce its adhesivity. That's inevitable as we age, and it's a cause of higher rates of retinal detachment in aging. That reduction in adhesion means that the holes that occur naturally from time to time can allow blood vessel penetration, and then you see blood vessel propagation, detachment, and failure of the retina. You can do a risk map where we ask the question, if I interfere with adhesion with the substrate, what happens? If I interfere with adhesion between retinal pigmented epithelial cells, which would be typical of what happens if I have inflammation in the eye, I actually get a different pattern of invasion, and that is seen clinically. If I have trauma, which interferes with photoreceptor RPE attachment, I can also get invasion, but the pattern is different. And again, that's seen clinically. And so we were able to explain why you see different patterns of retinal invasion under different circumstances, which the prevailing hypothesis of growth factors couldn't do. And in fact, later on, there were experiments done that validated that set of observations. Of course, when COVID hit, everyone wanted to study infection in one form or another. We did some basic studies, actually with a group of students through a hackathon, building a model of a tissue patch. Here are in blue, a group of lung epithelial cells. We start out with a single infected cell in the center. That cell releases virus, which infects other cells. It also releases cytokines that call in immune cells. The immune cells kill infected cells, also some uninfected cells. And you could build a little model of viral replication inside individual cells. And you could use a model of that kind to study, for example, what happens if you do antiviral treatment. The result here isn't earth-shaking. If you give a higher dose of the drug, the treatment's more effective. One of the problems with antivirals is that they have to be given very early. A viral load for a disease like flu peaks around the time you first notice that you're sick. You're maximally infectious before you know that you're sick. This happens also with COVID, although a little bit different pattern of viral load. And so this was a study of computational analog of remdesivir. We're going to interfere with RNA replication. Going from bottom to top, we have less interference at the bottom, higher interference at the top. On the left, we treat before we start the infection. On the right, we treat later and later. And as you might expect, if you treat early enough, you can get away with a low dose of the drug. If you treat later, you have to give a higher dose. And if you wait too long, no matter how much you give, it doesn't do anything. This is a classic pattern of antivirals, which is that they're great prophylactically. They're not so great once you have symptoms. You also see a lot of heterogeneity between individual replicas, which is seen in experimental clinically as well. So the one little exercise I was going to ask people to do in terms of coding was to go on to NanoHub and run that simulation. That simulation is implemented on NanoHub. If people could just take a minute or two to try this out. I will switch the screen share here to show the NanoHub version of this. Give me 1 second here. So let me get to the okay, so I'd like people to go to the NanoHub COVID CC3D COVID-19 tool. I'll put this in the chat. And see if people can get started on that. You're going to have 3 options here. K-on multiplier basically says how infectious the virus is between cells. So that basically represents the strength of binding between spike protein in COVID and ACE2 receptor. So if you go to started with alpha, that number would be pretty small, be higher in delta, and if you go to BA5, it would be bigger still. Beta delay multiplier represents the strength of the immune response. Higher value for that means that the immune system is slower. So a small value of beta delay multiplier would represent a young person. Higher value would represent an older person or somebody with immune suppression. And R max represents the maximal rate of viral replication. That probably doesn't change that much between strains of the virus, but a drug treatment with remdesivir specifically reduces R max. And so you can change that during the simulation, and that would be the equivalent of giving the drug treatment at that time. So I'll give everybody a minute just to run this. If you wanted to explore this, there's a lot to explore with it. I just wanted to let people try it out for a second or two. It does have a lot of windows. And so you may find that you have to do some rejigging. Remember that button under window, tile option here, tile, to lay things out again is useful. Remember, after you relay out the screen, you have to hit run again. And there are multiple fields that you could look at. There are 2 layers. I talked about a 2.5D model before. This is a 2-layer model, a top layer, which represents where the immune cells are moving, bottom layer 0, which represents the epithelial cells lining the lung. You can see the cells, you can see the virus field, you can see the cytokine or the oxidator that's released by the immune cells. So why don't I let people play with that just for a minute? If people are having trouble, let me know. I'll try a poll to see if that's working. If you really wanted to explore all the possibilities of this, it would take hours. So I'm not going to ask people to do all of that, just to get it right. TJ, if you're on the call, maybe you could just put the link to the paper, to the PLOS paper into the chat. Or Hayden, you might be able to do that.
Yeah, I don't know if I have that handy. I could go find it.
If you just go to Scholar and look me in, TJ. I have it, but I don't want to switch screens. So here I'm going to run it. Hope it'll run. There it goes. Boy, NanoHub is slow this morning. You'll see we have the amount of the number of cells of different type, how many cells have died, how many immune cells there are in the system, how much virus there is in the system. So you could visualize a lot of things. Again, this layout isn't the most elegant, but you can play with it and see how it works. So here
James?
is yes.
Yeah, we have
Pedro?
Which folder is this model in?
It's not. This is a NanoHub simulation. I put the link in the chat.
Yeah, I see. This is a embedded NanoHub. We recommend just following the link for this one and running through NanoHub.
Here you could see the red cell was the initially infected cell. The green cells are cells that have been infected by that initial cell. You're seeing a patch of infected cells around the initial cell, and that'll form a lesion. Actually, that's more typical of influenza lesions than of COVID. This was a model we built. This was actually a model that was written by a couple of undergraduates working with us in a hackathon in about 3 days back in April of 2020. And the paper was published in December of 2020. Relatively large paper. If anybody needs help, raise a hand. I don't want to take too much time on this, but I did want people to see a little bit of what you could do. Here you can see total diffusive species. You can see the population of virus is booming now. We're releasing, all those newly infected cells are releasing virus into the environment. And now probably we also have some immune cells coming in. Yeah, immune cells are beginning to come in as well. Layer 1 will show you the immune cells. So that's something I think you can play with. As I say, if you really want to explore that model completely, it takes quite a while, but you could do rather a lot. I'll warn you that for some settings of the parameters, you may have a crash in this particular model. We need to go back and clean that up a little bit. Another thing that you'll see here is the steering panel, which allows you to change parameter values in the simulation while it's running. We'll show you a little bit later how to write those. They're not perhaps as useful for research software, but if you want to distribute software, it's very nice to have this person running the simulation be able to change things in a controlled way. You should have gotten something that looks like this. Here I created some additional windows to visualize the immune cells, the virus field, and the cell field at the same time. So you can play with those as we go. We took that initial model, which was rather basic. It only had one immune cell type that aggregated CD8 plus T cells and NK cells in their behavior, only one cytokine. Used it, put it in a PBPK context, in a pharmacokinetic context, and tried to actually model the efficacy of remdesivir with more realistic parameters. Remdesivir is a prodrug, that is, the remdesivir itself is not active. It's a metabolite of remdesivir that's active. And that gives it a particular kinetic profile. And so we redid that naive diagram of a dose versus the time of initial treatment in more detail, looking at treatment efficacy and dose. And also something we studied, which I'm not going to show you here, was the effect of inhomogeneity. Not every cell in your body sees the same dose of the medicine. And that means that you can have some cells that aren't getting enough to turn off the viral replication. That happens a lot with cancer. You have a cytotoxic chemotherapy. It can kill 99% of the tumor cells, but if some of the tumor cells hide and escape, then the tumor will recur. All of those models used a very simplified model of the immune system. TJ, working with Barterman Trout and Erica Mohan, took a rather detailed model that had been well validated for influenza in mice and used this model that we see here on the right as a platform to actually build a detailed quantitative model of influenza infection in the tissue patch in the lung. And in this case, we've added the correct, not absolutely every species of immune cell, but quite a few more. CD4 plus T cells, B cells, antigen presenting cells, and that simplified single cytokine here has been broken up into a bunch of the main ones, TNF, IL-10, IL-12, type 1 and type 2 interferons, and antibodies, natural killer cells, and so on, macrophages. Still no neutrophils. Love to see somebody take that model and push it further. It'd be really pretty easy to take that and build an almost complete model of the immune response. Okay, so now I'm going to go back to the full PowerPoint. So we've also used this kind of modeling a lot in computational toxicology. We were very interested in liver toxicity. Acetaminophen, otherwise known as paracetamol in Europe, is medicine that's used for headaches, for fever. It can be pretty safe, but it also has the unpleasant property of causing liver failure in a significant number of people. And we did a series of experimental studies of liver failure in animals induced by acetaminophen. And we were interested in understanding transport of acetaminophen here in a single sinusoid in the liver. Here we're doing an explicit model of blood flow. In the middle, there's actually a CompuCell model of the flow of blood. It's expensive computationally, but you can do it. Here we have the hepatocytes on either side. Those hepatocytes are pulling acetaminophen, APAP, out of the blood. They're metabolizing it into its byproducts, particular toxic byproduct called NAPQI, NAPQI. NAPQI binds glutathione, which is a buffer of reactive oxygen species. We can fit this to clinical data on serum concentrations of acetaminophen and its metabolites. And we're able to actually replicate pretty accurately what happens when you take acetaminophen and its dosing in the liver. The liver has a complex flow pattern, and so this single pipe doesn't really give you a very good understanding of how the microdosimetry works. These are simulations of flow in the liver showing the distribution of acetaminophen exposure under different hypotheses. The Environmental Protection Agency has been using CompuCell for a long time, for more than 10 years, to study developmental toxicities. This was a study of Nicole Kleinspreyer's done back all the way in 2013, looking at vascular disruption effects from particular molecules. Max Long has been studying genital disruption by estrogen mimics, in particular feminization of male embryos by estrogen mimics. And Shane Hudson, who's going to be speaking to us on Thursday or Friday, did a beautiful model of secondary palate fusion of the bone structure development in the roof of the mouth, which can be impaired by chemical toxicants. In each one of these cases, there's quite a detailed molecular model of the signaling pathways. There also are what computational toxicologists would call adverse outcome pathways, which are network models which represent not only chemical species, but outcomes, cell death, cell proliferation, steatosis, and other things. And these are all CompuCell models, and they're beautiful examples of how you could build highly predictive, very specific and validated models of complex biomedical phenomena. And of course, in this case with the EPA, these are used in a generally regulatory context. So you need a high degree of fidelity in your models. So that, I hope, gives you a little bit of a sense of some of the things that you can do with a program like CompuCell. You could look at it in terms of basic biology, understanding development, homeostasis, developmental diseases. We can predict the effects of drugs and chemicals in certain cases, or toxicity, whether we're doing drug discovery or drug development or toxicology. I didn't really show much on the design of new therapies. The polycystic kidney disease is an example of real drug discovery approach. You could also try to do therapy optimization. And we've done some of that in the context of diabetic retinopathy. And in the interest of time today, I skipped those slides, although I'd be happy to talk about them another time. You'll notice that almost all the simulations that I showed involved cell movement or tissue reorganization. And multicellular models are particularly useful when you have cells proliferating and you want to know what's happening at the single cell, few cell level, or when you have cells migrating, for example, during metastasis. If you don't have any relative cell movement at all, then you can do larger scale simulations using continuum methods. And so, for example, heart and vascular modeling, where you don't typically have cells moving relative to each other, typically use continuum methods rather than the ones we do here in this class. But these things work together. And we did show you an example where we were doing explicit blood flow calculations. So I hope that gives people a little bit of a sense of the possibilities of this kind of modeling. And I'd like to move on to the other topic today, which may be a little bit abstract, but I hope will help you think about how to apply these things in your own work. And as I mentioned, one of the things that I've found over the many years I've worked in this area is that really the computation of the mathematics are the easiest part of modeling. The hardest part is deciding what you're going to model and what's a meaningful question. And so the things I'm going to talk about now may seem a little bit abstract, but I hope that they will be useful to you as you go forward. And I think having these in the back of your mind may help you process and integrate some of the things that we're going to cover in class. So the first thing, it maybe seems to beg and seems so obvious that it doesn't need to be said, but in my experience, nothing is ever obvious, at least until you've thought about it, is what is a model? And if we want to use a model, we have to have some way of comparing the predictions of the model to the real world. That means that we have to define a set of output metrics that we're going to use to ask the question, does the model in fact agree with the world or not? In biology, actually defining what those output metrics are is rather difficult. It's often the hardest part of the problem. The second thing is that a model takes two different inputs, one of which is a model structure that is fundamentally the set of biological, chemical, physical hypotheses that you're embodying in the model, and a set of input parameters. So I might have a model of a human heart, and one of my input parameters might be my blood pressure or my caffeine level. And this combination of model structure and parameters is something that requires some thought. We'll very casually talk about model structures and parameters, but in fact, we need to understand what those mean. A model structure basically embodies our scientific understanding of how processes work. Input parameters, like output metrics, can be problematic. A lot of times we can't measure the things that we care about. In the case of infectious disease, we would love to know in our output side what the viral load was in the patient as a function of time. If the viral load is measured once a day in the patient, you're lucky. It's almost never measured before the patient's hospitalized. That is during the period when the virus is beginning to replicate, the most important time for understanding what's going on. There are no measurements. If I want to quantify the immune system, I need a cytokine panel. You're lucky if you get those in a patient in a hospital once a week or once every 2 weeks. Cytokine levels can change every 15 minutes in a patient. So measuring them every 2 weeks doesn't tell you much. If you want to do immunomodulation, I'll tell you the data sets are not good. So you have to think about your own problem and ask the question, what could be measured on the input side? What could be measured on the output side? And what hypotheses do you want to embody? When we're going to talk about a model, this is now abstract terminology, but it's helpful to keep it back in your mind. If you're designing models, you need to do this. Absolutely. Is first, what are the things we're modeling? What are the objects we're going to model? Cells, extracellular matrix, chemical fields, what are they? We should always be asking why we're including. Then what are their properties or states? What level of detail are we going to use to describe them? We have to make decisions about that. What could we measure? What could we infer? Then we have to decide what they do, how they behave, and how they interact. That's the fun part, usually. We have to define their dynamics, how they change in time continually. And the difference between dynamics and events is continuous versus discrete processes. The growth of cells is dynamic. The division of a cell into 2 daughter cells is an event. It's a discrete process. We have to define initial conditions, how we start. Often that's not trivial. And boundary conditions, because we don't simulate the whole universe. We have to say what happens at the edge of our simulation domain. Boundary conditions would also include things like I give a drug at a given time. Always when you're thinking about building a model, or when you're thinking especially about a hackathon, define the objects you're going to be first, come back, and ask the question, what's the purpose of what you're doing? What's measurable on the input and output side? What are your hypotheses? And then, what are the things you're going to model and how do you describe them? And the more detailed you can be, the better. What do they do and how do they interact? How do they change in time? How do you start and what's at the edge? Everything has to be thought of in that way. If you can't do this analysis, you can't build a computational model, but more than that, you can't build a biological model because the same set of things that we're talking about here have to be done by a wet lab biologist to understand a problem. This is basic principles of modeling. It's not computing modeling or mathematical modeling. Then, if we actually want to build a computer model, there is a discipline for doing this. And one of the problems that we face very often is people try to short circuit it. People get the bit between their teeth and they want to run before they've done the basics. And I know it's frustrating. People want to code. But nothing leads to failure in modeling more than jumping into code without designing your problem. And I know that that can be a bit frustrating. And a hackathon, in a sense, is designed to push people into code fast, which is not necessarily the best approach. So there's a classic workflow of model development, which I'm not going to talk about in enormous detail, but I think is worth outlining a little bit. And I'll come back to the screen share. There's a always unfinished document that we sent out called Introduction and Template for Model Development Workflow that will walk you through these steps in a little bit more detail. So I'm going to go through this very quickly now, but if you want to go into more detail, we sent around by email a paper which you can look at on this topic. And the stages can be broken down in a lot of ways, but the most important one is to start out by defining your biological observations. What is it that you're trying to describe? Why is it interesting? What aspects of this problem are likely to be most significant? How would you measure them? And always coming back to the question of what's measurable, I think is very important. What did we use to decide what we thought was interesting? And also what aspects of the problem would allow us to control it? Because if all we can do is predict, we can learn scientifically, but fundamentally we can't advance medicine or engineering. Then you have to go through, and this is perhaps the single hardest part of model development, and define what your initial questions and hypotheses are. And very often the hardest thing is that we have ideas about relationships, but they're vague. What are the experimentally measurable things that we want our model to reproduce? To say that we want to reproduce cancer is not going to give us anything useful. To say that we want to reproduce metastasis by itself isn't going to say anything useful. If we say, what is the effect of changing substrate stiffness on the fraction of cells in a tumor that migrate out, that's beginning to be more a structured problem. What are the parameters that we could control in an experiment on the input side? What is their variation within a population? How do we generate a natural population that's meaningful? What are the scientific hypotheses under study? Again, the toxicologists with their idea of outcome pathways are very good at doing this. Just knowing molecular species doesn't give you much. You have to know how things function. What are the things that happen? And then we need to be very specific about the questions we want to ask. The most important thing then is to build what we call a conceptual or biological model. That is what biologists do naturally. It means drawing pictures, defining the components, and defining their relationships. And again, coming back to this diagram, we have to know what it is we're modeling, how we describe it, what its state or properties are. If we're going to have cells, we need to know where are they? What kind of cells do we have? What are the properties of those cells? How are they different from each other? Are the cells going through cell cycle? How do we define the cell cycle? How do we define its control? What are the behaviors we care about? There was a good question yesterday that when cells in an epithelium divide, they often round up, come out of the epithelium, divide, and then go back down into the epithelium. That's a very dramatic thing to see in an experiment. Having done a lot of experiments in chicken embryos, I've watched that a lot. But it may or may not be significant to the question you're trying to answer. If you're interested, for example, in midline extension, the fact that the cells in the lateral plate come out of the plane and come back down may or may not be important. And so you have to make decisions about what you think the key processes are, the key behaviors and interactions. You have to think about how they relate, dynamics, how do they control each other, change in time. And then again, conditions, initial conditions and boundary conditions. And so the more detailed your map of the conceptual model is, the more likely you are to build a simulation that's meaningful. And I think that that really is the hardest part. And I should say that this is not something that is a new, the best ideas perhaps are sometimes the oldest. When I had finished working this kind of schema out and talked about it, I picked up a book, ironically, a book on the history of theater of all things. And one of the chapters was on the Diderot-D'Alembert Encyclopedia from the 1780s. And in fact, the Diderot-D'Alembert Encyclopedia has a beautiful article on the importance of building conceptual models before building quantitative models. So the French already, at the time of the French Revolution, were making exactly this analogy here, this workflow. And it's ironic, perhaps a little bit, that this workflow, having been developed more than 200 years ago, still tends to get short-circuited. And the article in the encyclopedia also talks about why it's important to do it this way. So we have to learn from the greats from the past. Once you've built your conceptual model, you can go further. And I'd like people now just to take 5 minutes, and I'm just going to give you 5 minutes, precisely because it's enough time to begin to think about how hard the problem is. certainly not enough time to complete it. I'm sure everyone here has at least one biological problem that's of interest to them, maybe more. I'd like you to pick one biological problem, and I'd like you to begin to think about mapping it in the way we've talked about. And I will literally turn on an egg timer and have 5 minutes. I want you to think about what are the specific hypotheses you would like to test related to that problem. How could you measure the state of the system to determine whether you validated or falsified those hypotheses? What are the key components or objects that you want to model, include in the model, and why? What are the main processes, interactions, and dynamics you want to model? Why do you think they're important? And I'd like everybody to spend 5 minutes thinking about that in the context of your specific problem. And the reason I'd like you to do that now is because I think if you've done that today, as we go forward and teach methodologies and modeling components, you'll be able to much more clearly pull in and integrate the things that we're presenting as tools and ask the question, how do these work together? What we're doing together is learning a language. And language learning is always complicated because with no vocabulary, you can't say anything. But with no grammar and vocabulary, if you have vocabulary but no grammar, you can't organize. And so what we're doing here is trying to define the minimal vocabulary in terms of components, processes, and interactions, and the grammar, the minimum grammar. What is the basic set of hypotheses and relationships that we need to define our particular problem? Once we've done that, we can begin to say, okay, now we have a lecture on cell division. I know how to use that. We have a lecture on cell adhesion. I know how to use that. That's important to me. But until we think about a biological problem and ask why are these things important in my particular problem, all we're learning is empty vocabulary and empty grammar. So I'm going to give people 5 minutes to try out just working on that together.
I just want to tack onto that if you are working on a hackathon project idea, or if you're formulating one currently, it might be worth writing the answers to these down somewhere, because we can always use your thoughts on this to help scope your project if you feel like it might be a little too big right now during our open teaming sessions. So it's good to write these down and have them formalized somewhere as we move forward.
Yes, if you're going to do a hackathon, you'll have to go through this exercise. You may go through it formally on paper and write these down in the form of tables or diagrams. You may do it in your head informally, but ultimately to build any model, quantitative or qualitative, to understand the system, you have to go through this process. It's how our brains do it.
Last thing too, your answers to these questions will likely change as your project progresses. So they don't have to be perfect now. It's just the point is to get started thinking about them so you have something to work on.
Okay, I realize that there's no way one could do all of that in 5 minutes, but I hope it gives you something to think about. I showed you those simulations of choroidal neovascularization of the retina subsequent to age-related macular degeneration. One of the things that we lack actually are really good computational tools for helping you build conceptual models. It would be wonderful if there were great software tools that allowed us to build and manipulate conceptual models. We don't have them. Typically, we build things in the form of tables or figures. Biologists like figures. I like figures very much, diagrams. But sometimes you want to organize these things as tables too. And I just wanted to show you from that paper I talked about choroidal neovascularization. This was a formalization of the object types here, cells and fields, endothelial cells, photoreceptors, extracellular matrix, vascular cells, stalk cells, tip cells, RPE cells, and so on. And then a list of the things that they were doing, of their behaviors and interaction. And so this is the kind of thing that one would need to do formally to actually build a model, whether that is a conceptual model that you're going to be using to design experiments or a computational model that you're going to run on a computer. And so I thought you might like to see what that kind of thing looks like. when it's achieved in a published paper, in this case, this paper on blood vessels. So coming back to this, the next thing, once you've mapped what your components are, is that you have to begin to build a mathematical description of it, a quantitative description. You've said that your cells are going to migrate. Now you have to say that they migrate with a speed. How long do they move in a particular direction? What are the correlations? What are the metrics you're going to use to quantify the things that you've described? You're going to describe how fast your cells grow. You need a rate law for cell growth. If you have a rate law for cell growth, you have to decide what controls it. If you have a rate of cell death, you're going to have to decide what controls it. And defining those quantitative relationships is going to be the critical step in translating that structure, that model structure that you defined as your qualitative or conceptual biological model, into something that can be computed. Once you've done that, once you've done the definition of those structures and relationships, actually choosing a methodology for problem solution, whether it's CompuCell, PhysiCell, Biocellian, or something else, is relatively trivial. It's not that you can't make mistakes. It's not that you don't have to learn it. But the hardest part of doing biological modeling is the biology, and understanding the biology in a serious way, and understanding relationships in a serious way. And so this whole class is going to be focused on mathematical model implementing a simulation. And today is the one day we're really going to focus on, this morning, on thinking about the problems more holistically. And again, I can't tell you how often people start coding before they've decided what it is they want to represent. And that never works. It never works. The appropriate methods you're going to use computationally will depend, of course, on the problems you're asking. And you always want to use the simplest modeling approach that will suffice for the question at hand. If you could get away without modeling the spatial relationships of your system, then you shouldn't build a spatial model. And people like Alan Perlson have done tremendously valuable work on modeling diseases like HIV and designing new HIV therapies without modeling the spatial relationship within the body. If your cells don't need to move, then probably you don't need a multicellular model of the kind that we have. Simpler models have fewer parameters. They're easier to identify. They're faster to run. They're easier to understand. So you should always ask the question, if you go to a more complex model, if you add something to a model, is it needed? Is it essential? In the case of the remdesivir treatment model, there are beautiful papers by Goyal et al., for example. Lots of people from Seattle have done papers on remdesivir, which were non-spatial models. However, they couldn't handle the fact that you get different doses of the drug in different cells. Different cells in your body are exposed to different levels of virus. And so there was, in fact, a reason to go to a spatial model because the predictions would be different. And TJ has a nice paper where he takes some ODE models, spatializes them, shows that the parameters carry over exactly, but then that the predictions are different because the spatial inhomogeneity means that the ODE model mispredicts things. And so you have to be very careful. The other reason not to use the simplest model is that the simplest model may work for a particular set of circumstances, but when you go outside of that set of circumstances, it may not work. If a mechanism is important under certain conditions, and you don't include it in your model can't predict those outcomes. And this is actually the classic problem with artificial intelligence. Artificial intelligence depends on training data sets. If those training data sets come from the real world, by definition, they do not include things that don't happen. On the other hand, very often we want to predict what happens under circumstances we haven't seen before. AI typically cannot do that. Mechanistic models can do that if they're designed right. And that gives us a power that you don't get with AI methods. Now you could use the mechanistic models to generate training data for AI. So putting the two things together is the best of both worlds. But it's something to remember in this day of AI hype, that in fact AI has great limitations that are not usually discussed. So let's see. I'm going to talk a little bit about thinking about tissues from a virtual tissue perspective, and I'll run out of time, and we'll stop when we stop. I want to start out by just going over some very basic biology that will be familiar to everybody. And perhaps we can think about some of the questions we could ask about systems as we want to analyze those systems to begin to build models. If we start out at the highest level and ask what are the components of living tissues, well, almost always, in biology, there are always counterexamples. So if I deal with the acellular slime molds, the true slime molds, well, and I don't have cells. And we certainly have situations where we have syncytial organization, biofibers in the body, the chicken yolk, the Drosophila, early embryo. So cellular organization isn't absolutely universal in biology, but it's pretty useful. And as I mentioned, we're typically going to use cells as our agents in our models. We can model extracellular matrix. It's typically the hardest thing to model in our world if we want to model all of its details. The fiber architecture of ECM is really a problem for computation. That's not a problem for CompuCell, it's a problem for computation generally. And we have fluids. If we think about cells, the first thing we want to talk about are cell types. And cell type is a rather elastic concept. Two cells of the same type don't necessarily behave in the same way. They certainly don't have the same parameterizations. The number of cell types is not very well defined. On the other hand, the cell type name is something that's very useful. And one thing that we're going to face doing computation is that concepts that are intrinsically fuzzy biologically have to be mapped onto logically discrete concepts in our computation. In the real world, we might have two cell types that we think of as extreme, and if we did a flow cytometry, we'd see two clusters of cells around those central cell types, but we'd see all sorts of cells which have intermediate properties. Computationally, we have to give those cells names, and those names are Boolean. We either have that name or we don't. And so we're going to have to take fuzzy concepts biologically and draw barriers to cut them up when we build computer simulations. And that sometimes is not a problem. Sometimes it is a problem. The way we do those definitions to some extent is arbitrary. I can define, for example, a concept of a transport vehicle. And that transport vehicle could include automobiles, bicycles, buses, horses, maybe. And so I have a generic concept, which I'll call a type, which is a transport vehicle. I could define a transport type, a vehicle type of car versus a transport vehicle type of bicycle. Have that categorization. I could define within car, I could define types of Toyota, Camry, Oldsmobile, and so on. And I could define within those categories 2020 Toyota Camry, 2021 Toyota Camry, and so on. And so the way I define my names will define a little bit how I think about the problem. It's not that you can't switch when you're doing that, but it is actually does have a big effect on the way you think about problems. So type classification is actually tricky, and it's the first step for thinking about how to organize. Once we have decided what we're going to call ourselves, we have to make some basic assumptions. We have to say, how big are they? What's the range of sizes? What controls their sizes? If we're modeling things in 2D, we don't see how thick they are. What does that mean? If we have a columnar cell versus a squamous cell, what does that mean? We have to think about size scales. Cell shape is hard. CompuCell lets you have some degree of cell shape. If you really want complicated cell shapes, you can do it, but it's expensive. In reality, we typically are going to be modeling cells as blobs, extended blobs. On the other hand, if we wanted a Purkinje cell in the nervous system, we're going to have a hard time modeling that. And that's not unique to CompuCell, it's just hard because of the complexity. If we go to small scales, the podocytes in the kidney are terrifying. They have probably the single most complex architecture of any cell in your body. You may never have heard of them. They do filtration of the kidneys. We really have no idea how they build those structures. We have relatively little idea how the dendrites and axons build, but we really have no idea how the podocytes scale. Notice the scale here. The branching that happens in a neuron is happening on scales of microns. In the podocytes, it's happening on a scale of 100th of that. So these are only a couple of molecular fibers across. Myofibers can be very, very long, complicated. Typically, we're going to simplify that, but you have to decide what the ranges of sizes are that you care about and what level of detail you need. A fundamental concept which we've already run into is polarization of cells. We're going to have to decide if we're going to be satisfied with a blob cell that's uniform everywhere, or do we want to represent the fact that different regions of the cell have different properties? Depending on the questions we're going to ask, the level of detail we're going to need to represent those things is different. In particular, if the cells are secreting or absorbing from a particular surface, that's often very important. If force generation is anisotropic, we have apical contraction, we have an asymmetric contraction. Vertex models are usually used for epithelial sheet models because you could have one side of your cell contract and another side expand. You could do that in CompuCell, but it takes a little bit more work to implement it. You have to think about a lot of these processes. Again, we're going to be making radical simplifications, so we have to decide what level we can live with for our particular problem. So you have to ask questions. Ranges of cell size, shape, polarization. What level of detail do I need? How do they change in time? And critically, if we're going to include them, and they're not going to be static, what regulates those changes? What regulation is cell autonomous depends on the cell, and what regulation depends on how the cell interacts with its environment. A key principle of biology is that cells only know their internal state and local signal. They have no long-range information, and they don't know that they're part of a multicellular organism. There's no teleology. The only thing a cell has is its current state and its current environment, and does something in response to that. If I move the cell to a different environment, it'll sense the new environment, but it doesn't know its context. Then we have to ask what cells do. And the key thing is to think about the specific things that they're doing that are relevant to the question we're trying to answer. We're typically going to neglect all of the supporting machinery of the cell, unless we're going to be doing things like studies of toxicology or drugs that are related to metabolism. Just the way when you're driving a car, you don't typically care about how the spark plugs are working or how the gasoline is being injected into the engine. You care about how the car drives. Typically, in our simulations, we're going to neglect all of the metabolic components that keep the cell alive. We can only model things that are changing in time. And so we have to think about autonomous housekeeping behaviors of cells: metabolism, growth, cell cycle, cell death, volume regulation and chemical homeostasis, and differentiation. And again, each one of those we have to break down into the question of, is it important during the times we're looking at Do we need, what level of detail do we need to model it at? And how is it controlled? There's a lot that's known molecularly about these phenomena, but how they're integrated to give us control is not well understood. And one of the biggest problems that we're going to face when we build models is that we have all these data sets about the level of expression of gene A, B, C, or D. We even know quite a bit about connections between them these days, although less than we'd like to. The problem is that we don't know how the level of molecule X affects the behaviors that are actually the important things about what cells are doing. We want to know, is the cell dying or not? In that particular case, P53 may help us out. Caspase-8 may help us out, but you could have caspase-8 present in the cell, but depending on where it is in the cell, you might have a cell that's dying or not. And so you have to be careful in not being seduced by infinite amounts of molecular detail in trying to understand how cells work. Unfortunately, having parts lists doesn't tell you how things work. And that's probably the single hardest part of building these models. Once we have thought about cells, we have to think about cells in the context of a tissue. And I'm going to run out of time here, so I'm trying to that may cut this off a little bit early. We have to think about how tissues maintain their integrity. That means that there have to be signals that tell cells when to divide, when not to divide. We have to have cells be able to replace each other after damage. We have to be able to rebuild the extracellular environment. We have to have cells die when they go to the wrong place. We have to have cells control differentiation. Each one of these circuits, and there are multiple circuits, combines intracellular cell autonomous phenomena with signals. Some of those signals are diffusible signals. Some of those are signals that are contact signals with other cells or the extracellular environment. They could be electrical signals. They could be mechanical signals, pressure, substrate stiffness, and so on. We have to think about what cells do and how they interact. One of the most important things cells do, and by Fred Gabor-Forgax and Stuart Newman's book on the subject, that starts out with the sentence, the most important thing cells do is stick to each other, because otherwise you'd all be unicellular organisms. That's a good joke, but it's also true. We started out our simulations by talking about cell adhesion. And understanding cell adhesion is one of the fundamental things that we need to do. And it's one of the things that's studied relatively little in modern biology. How do cells generate forces to change shape or to move? How do cells polarize? How do cells move? There are a lot of ways cells move. What do cells secrete and absorb? How do they signal through contact? These are the fundamental questions. Sorry about that. Boy, that startled me. I guess I had a video that had some sound that shouldn't be there. But you'll see the. Let me skip that one. I don't know how I managed to get that to happen. That's funny. Startled myself. How do cells move? And so in conclusion, let me give you 5 minutes to think about these problems, having woken myself up by that video. And you asked you to spend 5 minutes thinking now about, you wrote a list of the components of your model and the questions you wanted to ask, and maybe some of the ways you can measure it. I'd like you to go forward and spend 5 minutes thinking about, what do you think are the most important behaviors, interactions, and regulation processes in your model? For example, some things you might want to think about specifically, are there specific cell-cell interactions, adhesion, signaling, contact guidance, cell extracellular matrix interactions? Those could be cell responding to ECM, it could be cell secreting ECM, it could be cell destroying ECM with MMPs or something else, reorganizing ECM. What are the key mechanical interactions in your problem? How do cells move? Why? Who's generating the forces? How are they transduced? What are the key regulatory and signaling pathways in your problem? What do you know about them quantitatively? The answer is usually not much. How do those regulatory and signaling pathways in molecular species affect the things that actually matter, which is what the cells are doing? So I'm going to give people 5 minutes to think about that as a little exercise. And then we'll have 1 or 2 more slides, and we will then wrap up. Perhaps as a framework for thinking about this kind of problem, I could show again this picture from Abbas's paper on choroidal neovascularization, where he's drawn the different cell types. And here he's drawn the adhesion interactions between the cell types as the set of functional hypotheses he's going to use to analyze the causes of this disease. Now, you don't necessarily have to make an analysis in terms of cell adhesion, but this is just an example of analyzing cell adhesion, in this case, cell-cell and cell extracellular matrix interactions. There might be many other kinds of interaction you do. I realize that 5 minutes is nowhere near enough time to answer problems of this kind. I hope that some of you will present your results. If we were a smaller group and we were in person, I would go around the room and have people present their ideas. But unfortunately, in the way this workshop is scheduled, we don't have time to do that here. I hope maybe if you decide to do some lightning talks, some of these ideas that you're working on now can come across in those lightning talks. I hope that'll be helpful for people. When you're talking about ECM, you have to think about binding and presentation of fusible molecules, transmission of forces, support and guidance, signaling, reorganization. Reorganization of ECM by calming is really a hard problem. We have to think about how fluids work. We'll hear about diffusion and reaction. We won't do much on flow and transport that way. Pressure signaling and flow signaling to cells is important. That's always an interesting problem to work on. We're not going to have time to talk much about tissue organization. I'll just remind you that there are 2 fundamental organizations of cell types of cell tissues, mesenchyme and epithelium. Here we see mesenchyme underneath and a layered structure on the top. There are all sorts of complicated structures, but all of the complicated roles, branches, and so on in our 3-dimensional bodies are fundamentally built out of epithelia. Epithelial cells have very complex structures. We're typically going to neglect a lot of the complexity of epithelia. It's going to be important that we recognize what we've ignored when we study epithelia. We often will assume that there is a difference between apical, basal, and lateral surfaces of cells and adhesion. We're probably not going to model all the complex junctional structures, but we almost never model the detailed fiber architectures inside of cells. Intermediate filaments in particular are rather important and get neglected a lot. We've talked a little bit about planar polarity. If you're going to talk about epithelia, you're going to have to understand planar polarity. That's a fascinating problem in its own right. And when we think about tissues, we have to think also about how cells organize at larger scales. How do we form sheets? How do we form clumps? How do we form tubes? How do tubes grow and branch? How do we organize within tissues? There are a whole variety of basic large-scale processes that are generated by the behavior of individual cells. And one fascinating question that we have to address is how do we get tissue-level reorganization and structure from cell-level behaviors? Cells don't have roadmaps. Everything has to be generated locally. Once we built a tissue, how do we maintain the tissue? How do we balance cell birth and death? How do we maintain oxygenation and generate angiogenesis without inappropriate angiogenesis? These are all things that understanding their signaling is really tricky, but also important. These are the kinds of problems that our modeling can really address. There's this nice picture from one of Stuart Newman's papers showing some of the different mechanical changes that occur in cell tissue, and cells organized into tissues. We again have to remember that cells only know local information. How do you get global organization from local information? And so I think these are things that we would all have to address, and most of the problems we're going to be interested in. Tomorrow we'll start on biological networks, and we'll talk a little bit about the four main kinds of biological networks: chemical reaction networks, and metabolic networks, signaling networks, gene regulatory networks, and transport networks. So those are all things that we're going to work on. And that's the end of my lecture. I'm going to give you the module poll. So please fill out your responses for that for today. And if there are any questions that people would like to have briefly in the chat, let me know and we can see about answering it. I'll give you 4 minutes to answer the module poll. Did the poll not start? I'm sorry. There we go. Thank you. Here I was looking at the poll saying, why isn't anyone answering it? The answer was I hadn't hit the right button. If you see a problem, please speak up, because the person giving the talk often doesn't know. There's nothing more embarrassing. Well, there are plenty of things that are embarrassing, but one thing that is quite embarrassing is to speak for half an hour and then be told that the slide share wasn't working. I suspect many of us have had that experience. So please do fill out your responses to the module, and I'll give people a minute for that. Again, if people have any questions, feel free to ask. So there was a question about how to make cells move faster on top of ECM. And I think the fundamental question there is going to come back to the biology of what you think is happening. Cells often move more rapidly when they're on a more rigid substrate, for example. And if that's the case, if that's the thing that you think is fundamental, then you probably have to include some details about how cells are binding to ECM. On the other hand, if it's just if you have an experimental observation that a cell of one type is moving faster than a cell of another type, then you can put that in by hand. You could say cells of a given type move at a particular speed, and then you can import that. And we'll show tomorrow, maybe later today, depending on how far we get, we will show you how to do variable cell speeds. So I hope that that answers that question a little bit. Other questions?
One way that we can make to cells move faster is applying forces, random forces, if you want the cell to move randomly. And also lowering the lambda volume and lowering contact energies also help the cell moving faster.
Pedro, you're in the room twice. Do you have your phone connected as well?
Any other questions? Okay. All right, let's see. So making contact energy lower might change the shape of the cell at the expense of, so again, things like contact energies, you need to think about in two ways. One of which is the shapes of cells are going to depend on relative contact energies, not on absolute contact energy. One of the things that we're going to do typically is we're going to assume that cells bind to each other. The contact energies that we're going to use are typically going to be positive. But in fact, adhesion is a negative contact energy. And the reason we use positive ones is if we use positive contact energies, we don't have to separately control the cell surface area. But in fact, it's probably technically more correct to include the cell surface area as a separate constraint, because membrane and cell adhesion molecules are regulated separately by cells. Anything else? Are people done with the poll? Does anybody need more time to fill out the poll? Last call on the poll. Okay, I'm going to close the poll then for the moment. And I'm going to ask people then to get ready for the next session. Appreciate you coming back. And I will turn it over. Let's take we're a little bit behind. Let's start again at 12 o'clock, or shall we say, for our next session? Hayden, you're the MC, so you tell me.
Sorry, what was the suggested? Yeah, if we come back at 12, that'll be
12 o'clock. We'll take a 10-minute break.
that'll be good. So Ajay, if you wanted to take a quick look at the issue you were having with Slack.
Yeah, good idea. Thanks.
Do you mind if Ajay screen shares for a second? James, he's having an issue logging into the Slack.
I think part of the problem might have been I've done this course several times, and at one time I might have done it as with one email, and then last year I did it with a different email, and my Slack might be confused, or I don't know what it is.
Yeah, I don't know. This is a new Slack workspace, so it shouldn't care which email you use. You may have a stale link.
So when I copied and pasted, there was an informational PowerPoint which had the Slack link. I put that in Google. I went to Slack, and then it asked me to choose an email. And when I chose my Google email, it said that I can't use it for some reason. Sorry. It said I couldn't use it for some reason, and then the only other choice was if I was at Indiana University. So
Where'd you say you got the link?
from the PowerPoint, the informational PowerPoint that was in the student materials. There's one slide which is a whole bunch of information and the orientation or something like that.
So I put another one in the chat. Try that one. You may have had the old link. They expire after 30 days.
Oh, I see.
So that may have been from an old PowerPoint. If anybody else is having the same problem, please let me know. Because if you have a link from more than 30 days ago, you can only join if you have an IU account. Because of the way Slack does it, it cancels it out. So I guess people can't find the link a year later and try to join and spam the workspace or whatever.
It's asking me to create an account, but I should have one. I'm just going to click it anyway. Let's see what happens.
Okay, I just got a notice saying you joined, so it at least
Okay, good.
is pulling you in.
Yeah.
Hopefully you see that on your end and it's not just mysteriously connecting you. to the server.
You're in. Welcome to Slack. Let's do it. And oh, here we are. Got it. CompuCell. Yeah.
Great.
Thank you.
Yep.